In this lecture, I'm going to discuss that how ATP synthesis takes place. The chemiosmotic model, which was proposed by Peter Mitchell, it explains that how the concentration gradient of protons is transformed into ATP. According to the model, the electrochemical energy, which is inherent in the difference in proton concentration and the separation of charge across the inner mitochondrial membrane, that's the proton motive force, it drives the synthesis of ATP as protons they flow passively back into the matrix through a proton pore in ATP synthase. Although the ATP synthase equilibrates ATP with ADP plus Pi, in the absence of proton gradient, the newly synthesized ATP, it doesn't leave the surface of the enzyme. It is the proton gradient that causes the enzyme to release the ATP formed on its surface. Mitochondrial ATP synthase is an F-type ATPase, which is similar in structure and mechanism to the ATP synthesis of chloroplasts and bacteria. This large enzyme complex of the inner mitochondrial membrane, it catalyzes the formation of ATP from ADP and Pi, driven by the flow of protons from the positive to the negative side of the membrane. ATP synthase, also called as the complex 5, it has two distinct components, that's F1, a peripheral membrane protein, and FO, which is an integral to the membrane. F1, the first factor which was recognized as essential for oxidative phosphorylation, was identified and purified by Ephraim Racker early in 1960. Mitochondrial F1, it has 9 subunits of 5 different types with the composition of alpha 3, 3 subunits of beta, gamma, delta, and eta. Each of the 3 beta subunits has one catalytic site for ATP synthesis. The knob-like portion of F1 is a flattened sphere which is about 8 nanometer high and 10 nanometers across, consisting of alternating alpha and beta subunits which are arranged like the sections of an orange. The polypeptides that make up the stalk of the F1 crystal structure, they are asymmetrically arranged with one domain of the single gamma subunit making up a central shaft that passes through F1 and another domain of gamma associated primarily with one of the 3 beta subunits designated beta empty. Although the amino acid sequences of the 3 beta subunits, they are identical, but their conformations they differ, in part just because of their association with the gamma subunits with just one of the 3. Well, the conformation differences among beta subunits, it extends to differences in their ATP to ADP binding sites. The FO complex with its proton pore is composed of 3 subunits A, B, C in the proportion of AB2 and CN, AB2 and Cn, where N ranges from 8 to 15 in various organisms. In yeast, it's about 10. Subunit C is a small, very hydrophobic polypeptide consisting almost entirely of 2 transmembrane helices with a small loop extending from the matrix side of the membrane. And these 2 helices, they are roughly perpendicular to the plane of the membrane and arranged in 2 concentric circles. The inner circle is made up of the amino terminal helices of each subunit, and the outer circle, which is about 55 angstrom in diameter, it's made up of carboxy terminal helices. The C subunits in this C ring, they rotate together as a unit around an axis perpendicular to the membrane. The eta and gamma subunits of F1, they form a leg and a foot that projects from the bottom side of the F1 and stands firmly on the ring of the C subunits. The A subunit, it consists of several hydrophobic helices that span the membrane in close association with one of the C subunits in the C ring. Paul Boyer proposed a rotational catalysis which is key to the binding chain mechanism for ATP synthesis. In this mechanism, he explained that the three active sites of F1 take turns catalyzing ATP synthesis. A given beta subunit, it starts in the beta ADP conformation, which binds ADP and PI from the surrounding medium. The subunit now changes conformation, assuming the beta ATP form that tightly binds and stabilizes ATP, bringing about the ready equilibration of ADP plus PI with ATP on the enzyme surface. Finally, the subunit changes to the beta empty conformation, which has very low affinity for ATP, and the newly synthesized ATP, it leaves the enzyme surface. Another round of catalysis begins when this subunit again assumes the beta ADP form and binds ADP and PI. Well, the conformational changes is central to this mechanism, and they are driven by the passage of protons through the FO portion of ATP synthase. The streaming of protons through the FO pore, it causes the cylinder of C subunits and the attached gamma subunits to rotate about the long axis of gamma, which is perpendicular to the plane of the membrane. The gamma subunit, it passes through the center of the alpha 3 beta 3 spheroids, which is held stationary relative to the membrane surface by the B2 and delta subunit. With each rotation of 120 degrees, gamma comes into contact with the different beta subunit, and the contact forces that beta subunit into the beta empty conformation. Well, the three beta subunits, they interact in such a way that when one assumes a beta empty conformation, its neighbor to one side must assume the beta ADP form, and the other neighbor the beta ATP form. Thus, one complete rotation of the gamma subunit, it causes each beta subunit to cycle through all three of its possible conformations, and for each rotation, 3 ATP are synthesized and released from the enzyme surface. The delta subunit, it confers oligomycin sensitivity on the ATP synthase, and the eta subunit may serve to inhibit its ATPase activity under some circumstances. The FO subunit, which consists of one A subunit and two B subunits, which anchor the FO-F1 complex in the membrane, and they act as a stator, holding the alpha and beta subunits in place. FO, as we discussed, also includes the C ring made up of a number 8 to 15 depending on the species of identical C subunits, which are small and hydrophobic proteins. The C ring and the A subunit interact to provide a transmembrane path for protons. Each C subunit has an aspartate residue near the middle of the membrane, which can bind or give up a proton. In this or we can say that it undergoes protonation or deprotonation during the catalytic cycle of the ATP synthase. The A subunit of the FO complex of the ATP synthase, as we discussed, has two hydrophilic half channels for protons, one leading from the P side to the middle of the membrane and the another leading from the middle of the membrane to the N side. The individual C subunits in FO, they are arranged in a circle about a central core. Each C subunit has as we discussed, that it has a critical aspartate residue about midway across the membrane, which can donate or accept a proton. The A subunit has a positively charged arginine side chain that forms an electrostatic interaction with the negatively charged carboxylate of aspartate on the adjoining C subunit. This C subunit is initially positioned so that a proton that enters the half channel encounters and protonates the aspartate residue, weakening its interaction with arginine. The arginine side chain rotates towards the protonated aspartate residue in the next C subunit and displaces its carboxyl proton as a new electrostatic arginine aspartate interaction forms. The displaced proton, it moves through the second half channel in a subunit A and is released on the N side. Thus, the C subunit with this unprotonated aspartate residue, it moves so that its arginine aspartate pair, it faces the half channel on the P side and a second cycle begins. Proton entry, protonation of aspartate, arginine movement, and proton exits. The rotation of the ring, it occurs by a thermal motion and is effectively rusted. The orientation of the protein gradient, it dictates the direction of the proton flow and makes the rotation of the C ring as unidirectional. By the large difference in proton concentration across the membrane, the number of protons that must be transferred to produce one complete rotation of the C ring is equal to the number of C subunits in the ring. In animal mitochondria, this number is 8. In yeast mitochondria and in E. coli, it's 10. And the number of C subunits, it can range as high as 15 in the cyanobacteria. Although the primary role of the proton gradient in mitochondria is to furnish energy for the synthesis of ATP, but the proton motive force, it also drives several transport processes essential to oxidative phosphorylation. In binding chain model for ATP synthase, the F1 complex, which has 3 non-equivalent adenine nucleotide binding sites, one for each pair of alpha and beta subunits. At any given moment, one of these sites is the beta ATP conformation, that's which binds ATP tightly. The second conformation is in the beta ADP, in which ADP is loosely, that's also called as the loose binding conformation, and the third is in the beta empty conformation, which is also called as the very loose binding conformation. The proton motive force causes rotation of the central shaft, the gamma subunit, which comes into contact with each alpha beta subunit pair in succession. This produces a cooperative conformational change in which the beta ATP is converted to the beta empty conformation and ATP dissociates. The beta ADP site is converted to the beta ATP conformation, which promotes condensation of bound ADP plus Pi to form ATP. And the beta empty site becomes a beta ADP site, which loosely binds ADP plus Pi entering. This model is based on the experimental findings, which requires at least 2 of the 3 catalytic sites alternate in activity. ATP cannot be released from one site unless and until ADP and Pi, they are bound to the other. To summarize, the chemiosmotic theory, it states that the role of electron transfer in mitochondrial ATP synthesis is simply to pump protons to create the electrochemical potential of the proton motive force should be able to replace electron transfer in driving ATP synthesis. This was proved by an experiment in which an artificially imposed electron chemical gradient could drive ATP synthesis in the absence of an oxidizable substrate as electron donor. In this 2-step experiment, in the first part, they isolated mitochondria and incubated in a pH 9 buffer containing 0.1 molar KCl, and slow leakage of buffer and KCl into the mitochondria eventually brings the matrix into equilibrium with the surrounding medium, and no oxidizable substrate are present in this. The mitochondria are now separated from the pH 9 buffer and they are resuspended in pH 7 buffer, which contains valinomycin but no KCl. Well, the change in buffer creates a difference of 2 pH units across the inner mitochondrial membrane, and the outward flow of potassium, which is carried down its concentration gradient without a counterion, creates a charge balance across the membrane, that is matrix negative. This because of the valinomycin which has been added. The sum of the chemical potential provided by the pH difference and the electrical potential provided by the separation of charge is a proton motive force which is large enough to support ATP synthesis in the absence of the oxidizable substrate. This is how a concentration gradient of protons, they are transformed into ATP. The mitochondrial oxidative phosphorylation, therefore, it poses no thermodynamic problem. But what is the chemical mechanism that couples proton flux with phosphorylation? This was given by the chemiosmotic model which was proposed by Peter Michell.